Hello everybody, this is your lecture on the organization of the nervous system and some information on basic sensory physiology. Recall in the organization of the nervous system that in the central nervous system you have the brain and spinal cord. What we want to focus on in this particular lecture is some information about functions of the spinal cord and brain. So we'll start with the spinal cord. The spinal cord has two primary purposes. It's the pathway for information between the body and the brain, and it's the site of spinal reflex arcs. So you recall from anatomy that inside the spinal cord you have your gray matter and your white matter. The gray matter is where information is processed, where you have cell bodies and synapses for that processing. In the white matter you have myelinated axons for carrying information excuse me, to and from the brain and spinal cord. On the outside of the spinal cord you have the spinal nerves. The spinal nerves have these branches here called the dorsal roots and the ventral roots. The dorsal roots are the side where information comes from the spinal nerve into the spinal cord, and the ventral roots is the side here where information comes out of the central nervous system and into the spinal nerve. Attached to the dorsal root you have this bulge called the dorsal root ganglion. The dorsal root ganglion will contain the cell bodies of the sensory neurons that are carrying information into the spinal cord. The cell bodies for the neurons that are carrying information out of the spinal cord are either going to be somewhere in the brainstem or here in the gray matter of the spinal cord. Now, spinal reflex arcs, or reflex arcs in general, are adaptations to reduce reaction time. Now, the way that reflex arcs reduce reaction time is by reducing the number of synapses involved in the process of sensation and action. The reason that this helps to reduce reaction time is because the number of each synapse in a process is going to introduce a small time delay called synaptic delay, a delay of about half a millisecond. So if you have a small number of synapses, you can keep your delay time within milliseconds. However, if you start to have thousands or more synapses, this can significantly increase the time delay between detection of a stimulus and your ability to respond to it. So reflex arcs can come in many different types, and we'll actually talk a lot about different types of reflexes in a lab, but you can see an example, a simplified example of a reflex arc or spinal reflex arc here on your left. So in each reflex arc, what you're going to have is some type of receptor that's going to be on the dendrites of a sensory neuron. That sensory neuron in this case is what's called a muscle spindle. This is a receptor that detects the length of a muscle. When a muscle starts to lengthen too much, which is sort of what you're stimulating in this patellar reflex that you see here, when you tap the patellar tendon, sort of activates this receptor, makes it think that the muscle is stretching too much. This activates that receptor. The receptor will generate action potentials that will travel up through the dorsal root into the spinal cord, into the gray matter of the spinal cord, where this sensory neuron will synapse with a motor neuron that you see here in the ventral root of the ventral horn of the spinal cord. That motor neuron will send its axon out the ventral root of the spinal cord and to the effector organ, in this case the skeletal muscle. And in this case the motor neuron will cause the skeletal muscle to contract and shorten the muscle and sort of prevent damage from overstretching of the muscle. What you see in this particular example, simplified example of a reflex arc, is sort of the simplest situation, where you have one sensory neuron, a single synapse, and one motor neuron. This is what's called a monosynaptic reflex. Monosynaptic reflexes are very simple, and so they can be very fast. Other types of reflex arcs that you'll learn will have different receptors, but also you'll have what are called polysynaptic reflexes, where you will have an additional neuron in the central nervous system, or at least one additional neuron in the central nervous system that's involved in the reflex arc. This, of course, will increase the time delay, but it also adds some extra complexity which might be needed in some types of reflexes. Let's move on now to talking about the brain. Recall from anatomy that you have a couple of major brain regions. This sort of top portion of the brain here, that's nice and colorful. This is called the cerebrum. Deep inside the cerebrum, sort of this region that you see here in yellow, you have what's called the diencephalon. And then sort of below the diencephalon, this region here and the region that you see in green here, you have what's called the brain stem. And then sort of behind the brain stem and under the cerebrum, you have this region called the cerebellum. Now, inside the brainstem, if you go from spinal cord here up to the top of the brainstem where the brainstem meets the diencephalon, you have the medulla oblongata, the pons, and the midbrain. All of these regions are going to have gray matter and white matter. Recall that gray matter regions are going to be regions where you have cell bodies and synapses, and this is where information can be processed. Recall that the white matter is composed basically of myelinated axons, and all that's going to be going on there is information moving up and down the brainstem to and from regions of the brain and spinal cord. The gray matter that you're going to have in this region of the brain, in the brainstem, and also here in the diencephalon, will be in small condensed clusters called nuclei. So there will be very important nuclei in each of these regions, and each of these nuclei are going to be sort of processing centers for various types of functions. The medulla oblongata in particular has processing centers involved in the respiratory system. These are called the respiratory rhythmicity centers, and they're going to determine, they're going to generate the electrical signals, They're going to generate the electrical signals that determine your respiratory rate or your respiratory rhythm. You also have cardiovascular centers in the medulla oblongata, one called the vasomotor center, one called the cardiac acceleratory center, or the CAC, one called the cardiac inhibitory center, or the CIC. The vasomotor center is going to be a center that controls whether arterioles vasodilate or vasoconstrict. By doing this, they can help to control blood pressure. The cardiac acceleratory center can speed up the heart if it's activated. The cardiac inhibitory center, the CIC, can slow down the heart when it's stimulated. The CIC and the CAC can also be used in the process of regulating blood pressure. There are also some major relay centers in the medulla oblongata for particular types of information. So if you have some type of somatic sensory information that's supposed to go to a particular region of the brain, that information may actually be directed by nuclei in the medulla oblongata. Next is the pons. In the pons, we don't have much in the way of nuclei. There are really only two that we're going to discuss, called the pneumotaxic center and the apneustic center. These two centers are both respiratory centers, and they can modify the activity in the respiratory rhythmicity centers in the medulla oblongata. Finally, we have the midbrain. In the midbrain, the gray matter regions are actually going to be more like the gray matter regions we're going to talk about are going to be sort of back here in a region of the midbrain called the tectum. This region of the midbrain, you have what are called superior colliculi and inferior colliculi, essentially these bumps that you can see here and here. The superior colliculi contains gray matter regions that are involved in visual reflexes. You guys have all experienced the visual reflex. When something, say, is coming towards your face very rapidly, this might cause you to sort of duck and cover. This is what we call a visual reflex. The inferior colliculi down here is going to be involved in auditory reflexes. And again, you've all been involved in these auditory reflexes, where a loud noise occurs behind you, you weren't expecting it, and it may cause you to jump. This is an auditory reflex, and it's processed here in the inferior colliculi. Also, these two colliculi, the visual one and the auditory one, are going to be involved in sort of basic location processing. So your ability to process object location with your vision is processed in the superior colliculi. Your ability to process sound location is processed in the inferior colliculi. Throughout these three regions, you have an important nuclei region called the reticular formation, which is involved in many different things. But one of the things that's kind of important is this is kind of the alertness center. And so based on the activity in this reticular formation, you can have various levels of alertness, all the way from when you're sleeping to where you're totally up and alert because you're having a fight or flight response. Next is the diencephalon. The diencephalon has two regions that are important that are not really identified here, so follow my pointer if you can. So here, just above the midbrain, there's a region here. This is called the thalamus. Okay. Now, one of the things I refer to here is this W shape that you see right here. W right there. Inside that W, this is the hypothalamus. And then sort of hanging off the back side of the thalamus here, you also have the epithalamus, or the pineal gland. The thalamus here is important because it's another relay station, but a much more important relay station for many different types of sensory and motor information that's going to and from the cerebrum. So a good way to think of the function of the thalamus is to think of it like a post office. Basically what the thalamus does is it takes information that's coming up from the spinal cord and sort of analyzes the type of information and says, oh, okay, your sensory information, you're supposed to go to this region of the cerebrum. That'll process sensory, or I should say somatic sensory information, or your auditory information, you're supposed to go to the region of the cerebrum that processes auditory information. So in that way, it sort of gathers sensory information and sorts it and sends it to the right place, just like you would do in the post office. Okay. Next is the hypothalamus. Remember, that's the W-shaped region. Inside the W-shaped region here. Now, this is why I included this table, because what you can see here is that even though this is an extremely small region of the brain here, this hypothalamus, it can be involved in a number of different things. The hypothalamus is going to be a major integrating center in reflex arcs. So it's going to be involved in the regulation of water balance. It's going to be involved in eating and drinking behaviors. It's involved in body temperature regulation. The hypothalamus is also a part of the endocrine system. This is going to be sort of a link between the endocrine system and the nervous system. Because the hypothalamus produces hormones, and it's going to use these hormones to control a number of endocrine organs, a number of endocrine glands. You can see some of those in the list here. The anterior pituitary gland, okay, it controls um, a reproductive function, which is not listed. Oh, here we go. Regulates reproductive function, number 5. It will do that by by producing hormones that are going to control reproductive functions. Okay. It's also a major player in regulation of the autonomic nervous system, see listed here. It's involved in circadian rhythms and participates in the production of emotional behavior. And in that way, it's part of this other system here called the limbic system. The limbic system is going to be sort of a multi-structure system inside the brain. But the limbic system is going to be involved in the generation of emotions and emotional behavior. And a particular region of the limbic system called the hippocampus is going to play an important role in learning. Finally, you have the pineal gland, which is not labeled, but it's this little bump back here. The pineal gland will produce a hormone called melatonin. This is the hormone that sort of makes you sleepy. This is the reason why they sell melatonin in sleeping pills. But technically you produce your own. Typically you're going to produce this melatonin from the pineal gland when you're not receiving a lot of light stimulation, essentially when it's dark. And so this is sort of part of those natural circadian rhythms that the hypothalamus will be involved in as well. Finally, we have the different regions of the cerebrum. In the cerebrum, you have your frontal lobe, your parietal lobe, your occipital lobe here in green, and your temporal lobe here in orange. The frontal lobe is going to have a number of important gray matter regions. The gray matter in the cerebrum and in the cerebellum, which we'll talk about next, are outer layers, outer sort of sheets of tissue that are highly folded, and this is why you get these folds in these two particular regions. It's actually the outer layers of gray matter that you find here. So those outer layers of gray matter are going to be called cortex. And so you're going to have different regions of cortex in these different lobes and in the cerebellum that are going to control various functions. So in the frontal lobe, you have the primary motor cortex. The primary motor cortex would be sort of back here, right next to the parietal lobe. This is going to be the region of cortex that generates the signals that make your muscles contract. So if you were to decide, I'm going to, do a biceps curl right now, that signal originally or originated in this region of the frontal lobe here. Also in the frontal lobe, kind of deep in this little crack here called the insula, you have the gustatory cortex. The gustatory cortex is going to be the region that processes information from your taste receptors and allows you to perceive taste. Now, something we haven't really discussed too much yet is this difference between sensation and perception. Sensation is really just the ability of your receptors to detect some particular stimuli. Sensation happens all the time. Your receptors are detecting things all the time. Perception is when that information ends up in these regions here in the cerebrum and you become aware of those sensations. So technically you can activate some taste receptors, but if you don't send that information to the gustatory cortex here in the frontal lobe, you do not have any perception. You're not aware of it, you don't know what's happening. Okay? Also in the frontal lobe, you have this region called the prefrontal cortex. The prefrontal cortex is going to be involved in motivation, in higher order functions like critical thinking, predicting consequences, and other things of this nature. Next is the parietal lobe. Now, they don't label it for you here, but you've got the dark purple and the light purple. These two regions are both parietal lobe. However, this dark purple region that they seem to be pointing to here for parietal lobe is actually a region of cortex in the parietal lobe called the primary somatosensory cortex. The primary somatosensory cortex is going to be the region that processes and allows you to perceive somatic senses. So remember that your somatic senses are going to be the senses from the body wall, like touch and pain and pressure and vibration and temperature. So all of those sensations are processed and perceived here. That means take pain, for example. This is the reason that you can sort of have some types of painkillers because when there's tissue, even when there's tissue damage, because the pain receptors are going to be detecting this tissue damage, and they're going to be sending the information to this region of the parietal lobe here. You're only going to be able to perceive that pain, to know that it's happening, if that information reaches the primary somatosensory cortex. So the idea with some painkillers is if we don't allow the information to get all the way up here, you won't really perceive that pain. But this is also a place where you would localize pain. This processing up here sort of helps you to determine where that pain is coming from or where the touch is coming from. All of that is processed in this primary somatosensory cortex. Next is going to be the occipital lobe. In the occipital lobe here, you have the visual cortex. The visual cortex, of course, is going to process visual information from the retina. Finally, here in the temporal lobe, you have the auditory cortex, which would be around here. The auditory cortex is going to process information from your sound receptors. And you also have the olfactory cortex in the temporal lobe. This is going to be the region of cortex that processes and perceives smell, information from your olfactory receptors. Finally, talking about the cerebellum. So the cerebellum is organized very much in the same way as the cerebrum. It's got this outer layer of cortex, of gray matter, called cerebellar cortex. And that outer layer of cerebellar cortex is going to be involved with coordinating movements. So there's lots and lots of sensory information that's gathered by the cerebellum, and it uses that sensory information to help you sort of make better movements, right? So when you're walking up a set of stairs, you have to make sure you lift your right leg to get up over the stair without lifting up your left leg, but you have to know exactly how high that stair is, where to place your foot, how your knee is bent, all kinds of different things all at once. What the cerebellum is going to do is help you gather and make sense of that information so that you can sort of make the proper movement. Lack of coordination would cause you to, produce these uncoordinated movements where you're tripping over yourself and such. In fact, one of the interesting things is that in studies people have found that when somebody drinks too much alcohol, alcohol actually stimulates the release of GABA in the cerebellum. Remember that GABA is an inhibitory neurotransmitter, and so it can inhibit some of this coordination activity that's going on in the cerebellum. And this will play a role in that lack of coordination that people have when they drink too much. Also, the cerebellum is going to be involved in some forms of learning, particularly motor types of learning, when you're learning motor tasks, how to do things requiring movement. What we want to move on to next is to look a little bit at some of the general features of our two divisions. Remember our motor or efferent division here, and the sensory or afferent division here. So we just want to look at generally how the neurons are organized and what neurotransmitters are used in these particular divisions. So this table actually will take your two motor divisions, which are going to be controlling effectors, and it will sort of give you, in this nice little table, some differences between the somatic motor division and the autonomic motor division. Remember that the somatic motor division is going to consist of these neurons that are going to control skeletal muscle. These somatic motor neurons are going to send their axons from the central nervous system all the way to the skeletal muscle cell itself. So what you'll see in the next image here is that in the somatic nervous system there will be a single neuron from the central nervous system to the muscle cell. When this neuron, this somatic motor neuron, innervates that skeletal muscle, it can only excite it. It cannot inhibit it. There's no such thing as an inhibitory somatic motor neuron. The autonomic nervous system, in contrast, actually uses two neurons to go from the central nervous system to the effector organ. Now here we can't say skeletal muscle, right? Because the autonomic nervous system controls smooth muscle, cardiac muscle, and glands, and those can be in a number of different organs. So we have to be a little general and say effector organ here. But the big difference is you have two neurons from the CNS to the effector in the autonomic nervous system, whereas in the somatic nervous system there's only one neuron from the CNS to the effector organ. Now, the other major difference you can see here is that autonomic nervous system can either be excitatory or inhibitory, essentially depending on where the autonomic nervous system is, where it's innervating. Now, this diagram shows you very nicely what you need to know about the innervation of the somatic branch of the nervous system and the autonomic branch of the nervous system. Notice that basically we have this little box here that represents the CNS. You have a single neuron that goes from the CNS to the skeletal muscle here. Okay? Now, what this shows you is one of the important things you need to know. This little region here, where the motor neuron reaches the skeletal muscle, this is a very special synapse called the neuromuscular junction, or NMJ. We'll talk a little bit more about that when we get to skeletal muscle. But in this neuromuscular junction, the motor neuron, when it receives action potentials, is going to release the neurotransmitter acetylcholine. Acetylcholine is going to bind to the NACH receptors. The N stands for nicotinic, and the ACH stands for acetylcholine. So this is the nicotinic acetylcholine receptor. Now this nicotinic acetylcholine receptor is a ligand-gated cation channel. It primarily opens channels for sodium, though technically these channels are permeable also to potassium in small amounts. But the way you should think about the NACH receptors, this is a ligand-gated sodium channel. It's an acetylcholine-gated sodium channel. So what you can see is this is why this particular system is always excitatory. If I release the acetylcholine, that's going to open the sodium channel in these NACH receptors, and it's going to depolarize the muscle cells. It will never hyperpolarize the muscle cells. It's only capable of depolarizing. And depolarization is what will lead you to threshold and generate action potentials, as we learned before. Now, when you look at the autonomic nervous system, you have to actually separate into parasympathetic and sympathetic divisions. But in both divisions, notice that you have one neuron from the CNS, two neurons from the CNS to the target tissue here. Okay, whether you're looking at parasympathetic or sympathetic. In the parasympathetic branch, you have this neuron that goes from the CNS and forms a synapse with a second neuron inside the structure called the ganglion. So this neuron here that goes from the CNS to the ganglion is called the preganglionic neuron. So the preganglionic neuron right here. Now that preganglionic neuron is going to release the neurotransmitter acetylcholine. And inside the ganglion, that acetylcholine is going to bind to nicotinic acetylcholine receptors on this second neuron called the postganglionic neuron. Now, just like we saw before, the nicotinic acetylcholine receptor is excitatory because it's a ligand-gated sodium channel. So that's going to generate graded potentials in this postganglionic neuron and eventually action potentials in this postganglionic neuron. Now, when that postganglionic neuron gets to the target tissue, it will again release acetylcholine into this synapse between the postganglionic neuron here and the target tissue here. Now, the difference here is that the acetylcholine will not bind to a nicotinic acetylcholine receptor. Notice that this is an MACH receptor, not an NACH receptor. The MACH receptor is a muscarinic acetylcholine receptor. The muscarinic acetylcholine receptor is actually a G protein coupled receptor. Now, when we talked about G protein coupled receptors in lecture before, I said you sort of have to take it tissue by tissue what this receptor will do, because there are so many different things that these receptors can do. So basically what that means is here at the target tissue, you can't say whether it will be excitatory or inhibitory because it can be either. It can be either one depending on what target tissue you're looking at here. In the sympathetic division, you have, again, a preganglionic neuron and a postganglionic neuron. Sympathetic preganglionic neurons are typically very short, like you see in the image here. These preganglionic neurons are going to release acetylcholine, and that acetylcholine is going to bind to nicotinic acetylcholine receptors on the postganglionic neuron. That will generate action potentials that travel down the axon of this postganglionic neuron and cause the release of norepinephrine. That's what the NE here stands for, norepinephrine, into the synapse here at the target tissue. Now, this norepinephrine is going to bind to receptors on the target tissue called adrenergic receptors. Adrenergic receptors come in two types. Alpha adrenergic receptors, which you'll find primarily in the smooth muscle in blood vessels, and beta adrenergic receptors, which we'll talk about in the heart and in the smooth muscle in your airways. So both the alpha and the beta, for our purposes, are going to bind to norepinephrine the same. Basically, you might learn differently if you take pharmacology or you take more advanced courses, but for our purposes, norepinephrine doesn't really distinguish between alpha and beta adrenergic receptors. Now, these adrenergic receptors, the alphas and the betas, on the target tissue are also G protein coupled receptors. So again, because they're G protein coupled receptors, we can't say whether they're going to be excitatory or inhibitory. We have to sort of take it on a case-by-case basis. The other thing about the sympathetic division, some of these preganglionic neurons do not go to a ganglia in outside the CNS. They actually go to structures inside the adrenal gland region called the adrenal medulla, and here they stimulate near what are called neuroendocrine cells, called chromaffin cells. These chromaffin cells, when they're stimulated by this preganglionic neuron from the sympathetic nervous system, Instead of releasing a neurotransmitter, they release a hormone into the bloodstream. This hormone is called epinephrine. Remember that the common name for epinephrine is adrenaline. And this is where these receptors that epinephrine and norepinephrine bind to, this is where these receptors get their name, right? Adrenergic sounds like a lot more like adrenaline, right? The epinephrine will bind to the same receptors as norepinephrine, the alpha adrenergic receptors and the beta adrenergic receptors on the target tissue. And again, as far as we're concerned, there isn't really a difference. The receptor doesn't distinguish between the two. But if you take more advanced classes like pharmacology classes, you'll learn that there are some differences. Finally, in this PowerPoint, we're going to talk about just some small amount of details about sensory systems. So one of the first things to talk about in sensory systems is generally what are sensory systems trying to do? What sensory systems are trying to do is take some type of external stimulus and convert that external stimulus into the language of the nervous system. If you were to take a light and shine it on the brain, is the brain going to be able to respond to a light? Hopefully your answer is no, because the brain doesn't have any receptors that can detect that light. Our receptors for light are in our eyes, and what those receptors do is they convert the stimulus that is light into action potentials, which the nervous system can process and understand. So that's essentially the goal of all of the sensory systems you talk about, to have some type of receptor that can convert this stimuli into action potentials. Now, in many cases, these receptors are on the dendrites of neurons like you see in this diagram here, but in some cases, those receptors are actually on specialized sensory receptor cells that will activate sensory neurons, which is the case that you see here. Now, once these sensory neurons gather this or get this electrical information, remember the processing and perception of that information occurs in the brain. It does not occur here at this level. So that means whatever activates this receptor is going to be perceived as the sensation that this pathway ultimately leads to. So let's say, for example, that this is a touch receptor here. Or let's say, for example, that this is a receptor for light in your eyes. If you press on your eyeball, that's going to basically produce some pressure in your eyeball. And sometimes these light receptors can actually be activated by pressure, not by light. Now, when that happens, you close your eyes, you press on your eyeball. What do you see? Do you perceive pressure? No, what you might see is you might see flashes of light. That's actually the perception of that pressure activating those light receptors. So this sort of idea is called the law of specific nerve energies. Basically what it states is that when you activate a particular type of receptor, like a light receptor, it doesn't matter what activates that receptor. What you're going to perceive is light. If you were, another good example would be a pain receptor. It doesn't matter whether it's acid or tissue damage or potassium that activates a pain receptor. What you're always going to perceive when you activate that pain receptor is pain. Now, what that also leads us to is if we have this situation where we have a bunch of different stimuli that can activate our receptors, then how do we know what the receptor is actually for? Well, it turns out that there are some stimuli that will activate these receptors better than others. And the stimulus that activates the receptor best is what we call the adequate stimulus. So for a photoreceptor or a light receptor, the adequate stimulus is light. Light is not the only stimulus, but it's the best stimulus. It's the one that activates the receptor best. This is a feature of sensory systems that we talked about in another context already. But basically, remember when you're talking about the nervous system, the way that you carry the strength of signal is in the number of action potentials generated. So they sort of show you this here with an example of from a touch receptor, where they activate this touch receptor with various intensities of touch. So this would be virtually no intensity here. This is a tiny bit of touch here, and this is sort of a major touch all the way over here. And notice that as you increase the strength of the stimulus, the number of action potentials that are generated increases because the signal strength is coded in the number of action potentials that are generated. Now, one of the things that's important to talk about with sensory systems is this idea of receptor adaptation. Because sensory receptors, some sensory receptors are known to rapidly adapt to a constant stimulus. Some receptors are known to slowly or never adapt to a constant stimulus. So you can see that here in the wording. The phasic receptors are receptors that are known to quickly adapt to a constant stimulus. Tonic receptors are receptors that slowly adapt, but that actually never stop responding to a constant stimulus. You can see that in the recordings here. So basically, this is the stimulus here, and notice that you're applying the stimulus for this entire length of time. But when you're looking at the phasic receptor, which is up here, you generate action potentials initially, then they start to fall off, and then they just stop altogether. This is a classic example of a phasic receptor. There are a lot of receptors that behave this way, and a lot of things that you may have noticed that come about from this. So classic examples of phasic receptors are touch receptors. This is the reason why you can wear a watch after a week or so. When you first put on that watch, you always notice it. It's kind of annoying, right? Because you're constantly activating the receptor, and the receptor in that case is responding. But after a while, after a week or so of wearing that watch, you may get to the point where you don't notice it anymore. Is the reason that you don't notice it because the receptor is not being activated anymore? No, the watch is always sort of stimulating that receptor. The difference is you don't notice it anymore because the receptor has just stopped responding to that constant stimulus. Another example of phasic receptor are olfactory receptors or smell receptors. And you may have noticed this when somebody walks into a room when they're wearing too much cologne or perfume. They walk into the room and the smell of the perfume or cologne is extremely strong. It might even give you a headache. But if you stay in that room with that individual long enough, eventually you won't even smell the cologne or perfume anymore. And the reason for that is not because the cologne or perfume disappears, but because the olfactory receptors have stopped responding to it. Classic examples of the tonic receptors, the ones that don't ever really stop responding, are nociceptors. Nociceptors are pain receptors, and they adapt slowly. As long as there's tissue damage or some type of painful stimulus, that pain receptor is going to respond to it. Finally, on our sensory lecture, I just want to introduce you to some of the general types of sensory receptors and the type of adequate stimuli that they respond to, and maybe just a little bit of information about each of these. So we'll start over here with photoreceptors. Photoreceptors are going to be the receptors that you have in the retina, in your eye. These are the receptors that respond to light. Their adequate stimulus is light. Now, there are two types of photoreceptors. There are rods and cones. Rods are going to be the photoreceptor cells that respond mostly to the intensity of light, and these are the receptors that can detect movement. Interestingly, rods are not capable of detecting the color of light. So technically, rod vision is black and white vision. Cones, on the other hand, are the photoreceptor cells that detect color. There are three types of cones: red cones, blue cones, and green cones. Each of these types of cones are sensitive to different wavelengths or different colors of light. Next are the mechanoreceptors. Mechanoreceptors are sensory receptors that respond primarily to manipulation of tissue or changing in the shape of tissue. So mechanoreceptors can be used for many different things. So touch receptors are mechanoreceptors. There are pressure receptors. Pressure receptors are what are known as baroreceptors. And typically these are blood pressure receptors that detect stretching of blood vessels. Also, you have what are called proprioceptors. Proprioceptors are receptors for joint and limb position. One experiment you should do while you're sitting there listening to this is close your eyes quickly and raise your arm up in the air. Even though that you can't see where your arm is, where say your elbow is, you know exactly where your elbow is. So then the question becomes, if you can't see where that elbow is, how do you know it's there? Because you have additional receptors in your joints called proprioceptors that will tell you the position of your joints at any position at any time. Next are the chemoreceptors. Chemoreceptors are going to be the receptors that respond to molecules. There are external chemoreceptors and internal chemoreceptors. The external chemoreceptors are your receptors for taste and your receptors for smell. So the taste receptors and the olfactory receptors, which are smell receptors. Taste receptors are going to detect molecules dissolved in saliva. Olfactory receptors are going to detect molecules in the air. You also have internal chemoreceptors uh, uh, for many different things. One of the major ones we'll encounter later are these chemoreceptors involved in regulating your breathing rate and breathing depth, called central and peripheral chemoreceptors that will detect carbon dioxide and pH and oxygen concentrations. Next are the nociceptors. Nociceptors are the technical term for pain receptors. And pain receptors are generalists. Pain receptors can respond to many different types of stimuli. Some that I mentioned to you before. Pain receptors can respond to tissue damage. They can respond to um, acids. There are some particular types of molecules that will activate pain receptors. Interestingly, one of those molecules is called capsaicin. Capsaicin is the active ingredient in chili. This is what makes chili hot. So technically, when you eat spicy food, what makes it spicy is the capsaicin activating pain receptors in your mouth, because they have that compound that in particular can activate pain receptors. Now, nociceptors are also unique because they're going to co-release the neurotransmitter glutamate and a neuropeptide called substance P when they send their information to the central nervous system. Now, that's kind of important, particularly for those of you who are going clinically, because there's a common drug that people use to inhibit pain that actually works by blocking the release of substance P from these neurons that are carrying pain information. That drug is called morphine. Morphine works by blocking the release of substance P from pain neurons. The last type of receptors are thermoreceptors. Thermoreceptors are receptors that detect temperature. Essentially, thermoreceptors come in two types. There are the uh, warm receptors and then what are called extreme receptors. Warm receptors are activated by sort of temperatures that are slightly higher than body temperature, but the extreme receptors are not going to respond unless the temperature goes way above body temperature or way below body temperature. Now, having these two types of receptors like this allows you to essentially process any type of a temperature stimuli. So let's say you have something that's very, very hot and it touches the surface of your skin. That will actually activate the warm receptors and the extreme receptors together. So if you have the two being activated, that will be perceived or processed in the brain as hot. However, if you touch something that's very cold, what that's going to do is it's going to activate only the extreme receptors. The warm receptors are not going to be activated by something that's very cold. And so when you have only extreme receptors being activated, this is perceived as cold. Now, this gives rise to this thing that you may have experienced before called paradoxical cold. When you go into the shower and you turn the shower on, if you turn the water on to extremely hot and you just quickly put your hand underneath the hot water, right away the first sensation that you feel might not actually be hot. It might be cold and then hot. This is because the information that travels on the warm receptors travels on slower axons than the information that travels on the extreme receptors. So when you first put your hand underneath the hot, what happens is the information from the extreme receptors gets to the brain first, and then after a short time delay, then you get the information from both the extreme and the warm. Remember that with a cold or with a stimuli where you activate only the extreme receptors, this is perceived as cold. So you'll perceive the cold first, and then when the warm receptors catch up, you'll realize all of a sudden, oh shoot, this is really hot.